Like I remember when I wrote a paper and I was like doing the graphs and I was like, this graph is 10 years or 10 months of my life. And you're just like, that's so depressing, but also kind of crazy at the same time.
How do you this? Man, I don't know how complicated this answer is going to be, but like, how do you genetically engineer or alter a mouse's DNA?
It's there's a whole lot of different ways you can do it. But what usually happens is you can like engineer the DNA in like the egg cell of a mouse and then implant it. So you basically like clone a mouse and engineer its DNA, and then you can breed it with other mice. So you get like the crossbreeding. So you can use a combination of like gene editing and selective breeding to like get a specific genotype you want. But then there's also cool things where like you can genetically engineer a mouse so that it has a normal genome basically until you give it something like environmental, like some exposure, and then it'll activate something that cuts out the gene. So like it can be totally normal until you give it like a certain antibiotic or you give it a certain drug.
How long until we can genetically engineer people to no longer be diabetic?
Well, type 1, we've already started to do that.
But type 1 and type 1 presumably we are doing that already because the DNA is already in there and we know
Well,
what to identify and what to change.
so basically, in most cases of type 1 diabetes, it's the result of an autoimmune disease. So they get some sort of event that happens where their body attacks the pancreas, the islet cells of their pancreas, the insulin producing cells of their pancreas, and it kills them. And so their body doesn't produce insulin anymore, right?